Hi, everyone. I would like to welcome you all to this month's webinar. And today we have a very exciting talk from Colm O Dushlaine. Colm is a data scientist with over 18 years of experience in computational biology and statistical genetics of large-scale datasets. He was formerly an associate director of statistical genetics and machine learning at the Regeneron Genetics Center, focused on integrating large-scale genetic datasets with electronic medical record information to find new drug targets. Prior to this, he was a senior computational biologist at the Broad Institute of MIT and Harvard, specializing in the genetics of psychiatric disease using association scans of array, exome, and genome sequence datasets. Colm received a BSc in biotechnology from Maynooth University, followed by a PhD from Royal College of Surgeons in Ireland. He gained postdoctoral experience at Trinity College Dublin and Massachusetts General Hospital in Boston, and he currently leads the Genomics and Data Science Group at 54Gene. The group is responsible for developing robust, scalable QC and analytical pipelines for sequence and array-based datasets and for driving target discovery efforts.
So yeah, thank you very much, and thank you so much for the invite. I wanted to use this presentation to give you a sense of what 54Gene are doing and how we plan to scale up our efforts across the continent and really bring precision medicine to to Africa, and by extension benefit the world through the new discoveries that we make given the increased diversity. So it's a incredibly exciting effort 54Gene are doing. It's and I think that I'm really looking forward to the wonderful discoveries and insights into populations that we hope to make. So so this won't come as a surprise. I think many of you have seen slides like this before, where a really tiny fraction of individuals in the GWAS catalog are African or of African descent, but they account for actually a disproportionate amount of modest or significant associations. And so there really is logic in pursuing more diverse populations, because that's where you hope to get more shots on goal. And but the vast majority of these are still incredibly small, underpowered, and we're really not sampling across a wide variety of African populations. These are really kind of localized, smaller studies. And so, really a kind of pan-African approach to GWAS and to increasing the, the number of populations across the country that we're sampling from will really increase the return on investment. So, on the right-hand side, we're seeing kind of the efforts that have been conducted to date, and it's incredibly modest when you really think about the scale of the continent and the number of the population of the continent, and we're only seeing about 5,000 to 10,000 genomes, versus like 1 million on a global scale. And less than 1% of the investment in genomics research and clinical infrastructure goes towards Africa. And so there really is kind of a call to action to dramatically increase this, to serve these underrepresented populations. So very high level introduction. So I'm going to talk a little bit about 54Gene. I'm going to talk about the Genomics and Data Science Group at 54Gene. And then I'll give you kind of just a really quick sneak peek into some of the early insights based on the initial sequencing of about 1,000 Nigerians that we've made on a kind of population genetics front. And then I'll also go over a custom array that we've designed that is specifically calibrated for African populations, and we hope to, use this low-cost array to really scale up our genotyping efforts across the continent. Oops. Okay, so I'll go briefly over this. PhD in bioinformatics, and then I postdoced, and you know, for about 5 or 6 years, I largely focused on psychiatric genetics. Psychiatric genetics are quite difficult and, you know, highly polygenic, and but it's a good training ground because you really get to dabble in lots of different techniques because it's very hard to find hits. So so that was a great experience. And then, you know, what we're seeing kind of in pharma are lots of you know, companies starting to invest in large-scale human genetics efforts, and you know, one of those is, you know, Decode Amgen, another one is the Regeneron Genetics Center, and really the idea here was to bring human genetic evidence to discoveries and to inform on you know, genes that are in their pipeline, and to bring additional evidence and support to gain approval. And then, you know, earlier this year, I moved to 54 Gene, and it's been a, you know, fantastic experience since then. Okay, and just to give you a sense of some of our, you know, folks who invest in us and some of our collaborators, it's really a, you know, a rapidly growing list, and some of those are from a, you know, technical perspective, some of those are from informatics perspective, and you know, we're featured in various, you know, news sources. And so, you know, we hope to continue to expand on this network of collaborations, some focusing on specific diseases, some focusing on infrastructure expansion, but it's been incredibly fruitful to date. A brief history of 54 Gene. It's a relatively new company. The idea emerged, you know, about 2018, and, you know, funding started coming in 2019, biobank installation, and things like that. And then in 2020 was really when, you know, the initial large injection of funding arrived, and we scaled up our labs and started commencing sequencing. And so towards the end of 2020, whole genome sequencing was, you know, well underway, and, you know, we have really good capacity to keep that up. And we also started, you know, into diagnostics infrastructure and molecular diagnostics. And then this year, we completed the design of a custom microarray. You know, we scaled up the data science group. We started to launch a translational biology team, which will bring a lot of support to, you know, to emerging candidates from our GWAS efforts. And we have started to expand beyond Nigeria towards other countries. And so, some of you may have seen, you know, this slide, and it's really just, and I'll, you know, kind of do this throughout the talk, driving home the message that there's, you know, significantly more, you know, absolute variation or even rare variation across, you know, all African countries, really, particularly in comparison to European countries, and so, you know, and more recently, and, you know, the H3 Africa effort, you know, they're able to characterize, you know, huge numbers of previously undescribed variants, you know, in their sample groups. You know, it's confounded somewhat by the fact that these populations haven't been studied, and so they would be undescribed. But it also would suggest that there is, you know, novel variation, you know, to be discovered in these populations, and that really helps drive target discovery, and also, you know, improve power to detect targets to begin with by having higher frequencies. Also, when it comes to rare diseases, having more, because they're so underpowered, you know, having a lot of rare diseases aggregate, a lot of rare variants aggregating at the level of a gene can give you kind of more combined power to detect associations in these kind of burden tests. And just one more slide on this, and, you know, you may have seen this as well. It's from the 1,000 Genomes paper showing, you know, country-specific and continent-specific variation, and we can see that, you know, typically in the small number of sample populations in Africa, we can see a huge amount of private variation. But by contrast, you know, European descent populations don't really, you know, have that many variants that are unique to those populations. And in this H3 Africa paper, you can see in Botswana, we can see a huge amount of novel variation. And while that could be just because the population hasn't been studied, it is perhaps more likely, in my view, that there's just lots of unique population-specific variants that, you know, arise for numerous reasons. You know, drift being one, but also the kind of the history of of disease or migration or admixture in that population. And then in this circus plot, this is from the TopMed consortium. I think what's interesting about this is if you look at singletons and rare variant sharing, you can see that in the African populations, the extent to which rare variants are shared across African groups is higher, and that is really interesting when you start thinking about exome sequencing or whole genome sequencing and identifying those rare variants, because you really want to be getting more copies of rare variants at a gene to find those really ultra rare protective mutations. And so again, that reinforces the notion that focusing on African populations makes complete sense. Why did we start in Nigeria? It's a huge population, with over 200 million people. There's huge tribal diversity, and actually it is remarkable to see, to compare that to kind of this vast swath of Africa where that combined represents the same population. And so one thing is sampling across the continent, but the other thing is sampling in a region where you would expect a lot of tribal diversity to aggregate by virtue of it being a highly dense kind of urban region. But we do also know that there are a lot of populations, and they arise in various parts of the country in greater or lesser extent. And actually, I'll touch on this again later in the talk. So pivoting back into 54Gene and some of its research infrastructure, we have capacity for genomics research, we have capacity for histopathology, we have large biobanking capacity, currently at about 300,000 samples minus 80, room temperature capacity of about 100,000. And we are expanding into molecular diagnostics, NIPT and various clinical diagnostics approaches. In terms of informatics capability, we're expanding our bioinformatics and statistical genetics groups. We have a huge amount of molecular genetic scientists, biorepository scientists, clinical microbiologists, and medical laboratory scientists. And to touch on some of the studies and some of the initiatives, and this is one that is in 20 tertiary hospitals. That covers about 8.5 million patients per year, 250 field researchers, 5 key disease areas right now, but we will continue to expand that, and moving towards an EMR-based framework where we can, kind of more digitize records in a more efficient way, and also, collate and collect more phenotypes, and not just kind of specific phenotypes. And so I think EMRs are particularly useful in that regard. We collect, a lot of, a reasonable amount of traits on these individuals, family history, comorbidities, household metrics, health behaviors, things like that. And then, within cancers and neurodegenerative and cardiovascular, we have kind of, stratified phenotypes and different kind of subgroups of the disease. Okay. That is a hospital-based cohort, which is useful in one sense, but it is enriched for can be enriched for sick individuals, and so by contrast, community-based collection is also incredibly useful. And this describes a door-to-door effort where we're interviewing families and trying to get members of multiple members of households, and that can be very useful for family-based analyses, and also describing maybe in a less biased way the prevalence of diseases once we start hitting large enough sample sizes. And so that continues to expand as well, and we're in about 500 communities and really all over the country, and currently 30,000 participants. So to kind of touch on the genomics and data science group and kind of where we fit, right? So we have phenotype collection, we have biobanking and genotyping and sequencing efforts, and then once those data are generated, our job is to work through that data. And in the case of phenotypes and genotypes, run GWAS, run target discovery efforts. But even before getting genotypes, cleaning and processing the raw sequence data so that it is up to scratch for various analyses. And then we also weigh in on on data collection and things like covariates and the structure of the phenotypes and the phenotypes looking clean, and also things like power calculations and what are relative power to detect various variants of certain frequencies are given the prevalence of the disease. But I'll touch on just one here, and so this is work led by Barry Ballou and my group, and the idea here is to develop a like a scalable, flexible solution that can work in multiple different environments, that can take raw outputs from the sequencer, and in our case, in a cloud-based and AWS framework, effectively bring that all the way through to genotypes that we would be prepared to analyze statistically, right? And so we've a Git repository that we will make public, with publications. We have automated jobs. We use things like spot instances, and we've kind of an intelligent pipeline in the sense that when jobs fail, we can ask the nodes to grab more, more RAM and kind of try again. And so that's a really cost-effective solution. And then we can scale up depending on the throughput in the lab, and we currently are calibrated for 100 whole genome sequences, but we can scale that up quite easily. And this will work in other platforms, and we hope that others do use it when we share it. And so just to go into this in a bit more detail, data comes off the sequencer. It is aligned. BAM files are generated. We do some post-alignment QC. Variants are called in each individual, and then across the whole cohort. So the single sample GVCFs are kind of put together and jointly genotyped. And then at the end of that, we've split multi-allelics, we apply some additional sample and variant quality filters, and we merge all the clean calls back together again. And so we have this, at the end of this, we have this multi-sample VCF file effectively. And then the last slide I'll show on this is, these are the outputs. So FASTQ goes in, and the outputs are, a VCF file with various, metrics and individuals that fail for various reasons, head-home ratios, low coverage, things like that. Accompanying that, and then we also produce BAM files, and then we produce a number of metrics, and these QC metrics are things we can tweak and we can iterate and we can explore as sequencing continues. How do these metrics look in other sites? If the sequencing technology changes, how do these metrics look? And this is very important for identifying batch effects or things that would get us a little bit worried about jointly analyzing the data. And so we might need to run a meta-analysis if we thought there were too many batch effects going on. And these are just examples of quality control plots where we kind of, you can see really, unusual outliers. And if we see a few of those, we typically remove those. We assess the missingness rate and filters according to, GATK best practices, things like quality by depth and such. And so, we started, whole genome sequencing in the first quarter of this year. The quality, is very high, and we don't see many dropouts. And so we're quite happy that we're future-proofed in that regard. Okay, so now pivoting towards PopGen a bit. This map represents tribes, reported in a census of Nigeria, I think in 2006. And what's really remarkable about this is, well, the amount of tribes we're seeing, but also, you know, the patterns, and some are localized, some are, you know, in a few different spots. And, you know, this is something that we're thinking a lot about as we look at how individuals, where individuals are falling out in principal component space, for example. And so we are trying to, you know, view our genetic data through the lens of kind of what is the actual, you know, distribution of tribes across the country. And obviously states don't equal to tribes, and that's something that, you know, we keep in mind as well. But before I kind of go into the genetic data, this is just an overview of where we're the states that we're sampling individuals from, and the size of the dots roughly, you know, correlate with the number of samples that we're taking or collecting in these regions. And the plot on the right, I'm showing, you know, state tribe combinations, and colors are by the you know, the number, you know, in bins of numbers of individuals, and so it could be, you know, 1 to 10, 10 to 100, 100 to 5,000, etc. And, you know, one thing that is worth noting here is, you know, one thing on one hand, you can try to collect across the country and make sure you're, you know, representing all tribes, but, you know, there are also regions in the country where we're seeing, you know, 150 unique tribes. And, you know, a lot of those are very low numbers, but, you know, it is still very much worth targeting specific, you know, cosmopolitan centers, and you will get, you know, that sample that tribal diversity. Okay, so moving into kind of principal components, and, you know, I'm guessing many on this call are familiar with this. This is kind of our first analysis of how individuals in our data compare to individuals in 1,000 genomes. And, you know, not surprisingly, our samples are very much African and consistent with African samples in 1,000 genomes. And, you know, these are African, and this is a mixture plot. These are samples, African samples in 1,000 genomes, African samples in our data. And, you know, diving in a bit deeper into the, you know, the structure locally, and this is work by Cameron Palmer and my group. You know, here we're comparing in blue 54 gene samples, and then these other colors are, you know, 1,000 genome samples. And so we can see in I think it's dark green, we can see Isan, and in orange we can see Aruba. And, you know, our samples overlap quite closely with samples, but also our tribes align quite closely with Yoruba, Isan tribes. We do sample from those groups, and we see them, they're very, very similar. So that's reassuring. And what I wanted to kind of step you through here is, you know, really just showing you that although the sample sizes are quite small for some of our tribes, we can really see different patterns, right? And so here's one tribe, and, you know, they're very tightly grouped. And if we step through, we can see another, you know, tribe that's very tightly grouped here. But there are other tribes in our data that cover much, you know, wider genetic distances, and that could be for a number of reasons. They may have been more nomadic, or they, you know, maybe just more highly admixed, or things like that. And so we're very interested in investigating these, you know, fully, and trying to figure out their histories a bit more. And just to, you know, to bring that a little bit further, you know, it could also represent, you know, the effects of, you know, introgression from Northern African or European populations. And so here I'm showing 1,000 genomes populations, and also, you know, Southern Italians, and as a proxy for North Africa / Europe. And we can see that some tribes, and, you know, these lines broadly, you know, separate out tribes in our data. You know, and we can see, you know, what might be a modest signal from you know, regions north of Africa in our data. So we're very interested in exploring, you know, to what extent, you know, we're seeing that admixture and introgression in you know, across different tribes in our data. And just to briefly using a higher K value for admixture, just to touch on some themes that we're seeing, right? If we use 1,000 genomes, African samples to kind of calibrate our expectations, we see this kind of this green representing a west and northwest pattern. We see kind of an eastern pattern, and so these are tribes in our data that might have kind of eastern like an eastern genetic component, and then we kind of what you could call like a broadly Nigerian pattern, which is represented in purple. But these are just incredibly early investigations, and we're still kind of working through a lot of the data. And also just touching really briefly on this, we don't see what you would classically call large runs of homozygosity in our data. There are many reasons why why runs homozygosities would differ, but at a very high level, even across all the different tribes, we see quite remarkable consistency and not much variation in kind of estimates of inbreeding, certainly in comparison to what you would see in European or South American populations from the 1,000 genomes. And so that's interesting, and we're curious to pursue that and refine that a bit more. Yeah, and then just to circle back to this H3 Africa plot, which I do find fascinating. That really these it is remarkable to see the variation in kind of yield of novel variants across populations, and I do think this will extend not only from countries all the way to tribes, because the uniqueness of the histories and behaviors of these tribes is profound, and I think having a representative sampling from all these tribes and countries is really critical. Okay, and so I'll pivot a little bit into things that we're starting to think about in terms of adverse drug reactions, and this is quite new to me. I only learned this recently, but the rates of adverse drug reactions and particularly fatalities in African hospitals is significantly higher than those in Europe. It's 6 times higher, and that is remarkable. It accounts for 16% of hospital deaths in Africa, and we could argue that disease burden, socioeconomic status factors, things like that could be at play, but we could also make some argument that genetic diversity and allele frequency differences and poor characterization of those things is also making a difference. And there are some studies that I'm just citing here. The cytochrome P450 enzymes have significantly higher diversity in Africa, and that hasn't been well characterized. And it hasn't been characterized to what extent these enzymes are affected by these variants and how that would affect the metabolism of drugs. Similarly, drug transporters can either reduce or increase binding to specific drugs, and they have higher frequencies in non-African populations. Another story, that the proportion of variation in warfarin response in Europeans is pretty well understood, with 3 mutations explaining kind of a significant amount of the variability in metabolism of the drug. But we don't know those modifying variants, and certainly these particular variants are at a lower frequency, so there may be many more modifiers that just haven't been well characterized. And then a note, story of LabDAP, which was kind of withdrawn from the market due to effectively a poor understanding of people who are at risk of hemolytic anemia and severe anemic reaction if they were already G6PD deficient. And these are, in theory, things that we could get a much better understanding of by really characterizing the allelic mutational spectrum of variants of these kind of well-known genes. And so really, in my view, there's a clear need to establish a catalog of variants that would modify drug efficacy and toxicity, and to implement screening procedures, if possible. And tying this into some of our data, these are just some selected pharmacogenomic SNPs, reported in the Genomes Asia 100K paper, and just a quick look. And these are reasonably small numbers, maybe 20 to 30 or 40 samples in the selected tribes. But really, we're showing the monoallele frequency across tribes on the X, and the monoallele frequency on the Y. And we can see that sometimes there's reasonable consistency, but sometimes we're seeing a lot of variation. And so for this variant, we're seeing a really high frequency in this tribe. And so that in terms of a precision medicine thing, it would be really good to to be able to leverage what we know about mutation rates in different tribes, and how that can be assigned to individual risk. And this is not dissimilar to H3 Africa reporting. We can see a lot of frequencies, and HBS, for example, is highly dependent on and coincident with the prevalence of malaria across the continent. So so I think these efforts are underway. They've been carried out in H3 Africa. We continue to carry them out in our own work, and I think over time we'll move towards a much more comprehensive catalog of these variants. So I'll touch on one more story where it just shows the power, in my view, and it shows the power of diverse populations and what they can bring to drug discovery, and in this case, drug discovery that will benefit all populations. It's really about these biological insights. It's not, these are not hits that are going to just benefit the countries where they were, or continents where they were discovered. So PCSK9 correlates with lower levels of certain blood lipids, but variation or protective variation in particular is extremely rare in Europeans, but relatively common in African Americans. So it was identified as a therapeutic target using evidence from two populations. Right? One was French, one was African American. In the French sample, if a founder population from Quebec, 2 gain-of-function mutations were characterized in families with familial hypercholesterolemia. And so, these individuals are very young. The gene is in overdrive. It's working harder than it should, and they're getting heart disease in their 20s and 30s. Loss of function mutations, the kind of dial down the expression of the gene or break the gene, tend to be associated with more favorable lipid profiles. And these were identified in African Americans in the Dallas Heart Study. And so, those two stories showing kind of protection and risk, gain of function, loss of function, you make a really compelling story. And PCSK9 inhibitors are now used in treatment of hypercholesterolemia, and it shows the advantage of, in this case, two diverse populations from kind of the bottlenecked end of the spectrum and the far more diverse end of the spectrum. They can make insights that can benefit global populations, and and even now, there are efforts to effectively make this a permanent therapy, where CRISPR is used to alter cells in the liver for kind of permanent breaking, I suppose, of PCSK9 in the liver. And then just to drive this home again, we've more in diverse populations. We have just far more shots on goal. We have more variation. We have more unique populations. We may well have a wider spectrum of a phenotype and kind of a wider distribution of the phenotype. And in founder populations, these kind of experiments of nature, we have just by chance, a founder population splits off, and the initial individuals have certain variants, and those variants will arise to higher frequency just due to the bottleneck. And there are lots of examples of just increased power to detect those really interesting variants just by chance. And so in the case of founders, the objective really is to sample from a lot of different founder groups, and that will kind of give you hopefully more shots on goal that way. So, you know, here's another example that's relevant to where we are now. You know, this is an example of a protective mutation that's, you know, that basically reduces the risk of people becoming seriously ill with COVID-19. And, you know, it's found at pretty high frequencies, you know, in many countries, more so in Asia. But we, you know, we don't see it at all actually in African groups. But we don't know, is that even true? We really do need, we're just not sampling appropriately from the continent. But this is an example in this case of a protective mutation, but there might be, you know, many examples of that. And that would also, in the case of COVID, for example, give us insights as we conduct, you know, GWAS studies during epidemics, for example. If there is a protective mutation that's specific to a population, and we have a lot of data on that population, we can run GWAS efforts to identify that mutation, and that will give us quicker insights into the gene in question and allow us to kind of, you know, develop therapeutics. So lastly, I'll touch on our efforts to develop a low-cost microarray. And so, you know, I talked about whole genome sequencing. That is really expensive and generates a lot of data. You know, an alternative, a kind of a first pass, is to run your samples through a microarray. And because we have whole genome sequence data, we can also use the whole genome sequence data that we've already generated as an imputation reference. And so it's, you know, really, it's a really nice complement to the whole genome sequence data to run this array. And so we designed it to, you know, try to be as well calibrated to the African continent as we could, given the data we had. So we used backbone content from you know, powered chip and mega arrays, really as the bulk of the content on this chip. Then we also spiked in custom content, you know, things that we believed in ClinVar, GWAS catalog variants that were African specific. So we didn't take everything from the GWAS catalog. We took things from, you know, African GWAS from the GWAS catalog, from GKB, EQTLs from specific the AFGR, African Functional Genomic Resource. We spiked in kind of approximately independent EQTLs from that study. And then markers that differentiated across some of our larger tribes and markers that were predicted to be deleterious in our whole genome sequencing data. And then, you know, some of the canonical variants of interest. So that's the chip, and that's work led by Cam and the group. So in the last few slides, you know, I'll just talk about how we are scaling up our efforts. And so we've been heavily focused on Nigeria, but we're moving in the soon future into Senegal, Kenya, and Tanzania. And, you know, again, while not these countries, while they don't have as many tribes, it appears, as Nigeria, they do have significant numbers of distinct tribes. And so we hope that can continue to add to our sampling of tribal diversity. And it will also help us identify, you know, novel variants that we might want to, you know, layer into the custom array as we, you know, develop that over time. Some of the future initiatives that I think are, you know, really exciting, and we're going to kind of carve out a lot of time to look at these. So number one would be looking at Neanderthal or ancient. We call this ghost. It's ghost because we don't really know what lineages, human lineages, extinct lineages these are. Ghosted mixture in African populations to identify events that might have conferred beneficial adaptations to populations. And so, you know, there's a lot of work has been done on Neanderthal, but even the extent of Neanderthal introgression into Africa hasn't really been well explored, or certainly well explored in a collection with you know, as large as ours, with sequencing as kind of as deep. So we also want to support the creation of kind of a Pan-African imputation reference variant catalog. And that will really help, you know, clinicians on the continent and researchers who are using microarrays to impute and really increase the number of variants that they get with their microarray. But they also allows clinicians to look up frequencies and not rely on European frequencies that might be very low and give you the impression that it's not a variant of interest. We really want frequencies that are relevant to the population that we're studying. We want to continue to evaluate the custom array performance with different reference panels and tweak it over time. We want to start doing a lot of different studies in kind of the rare disease space. We will start expanding the number of genome-wide association studies and probably eventually move into polygenic risk scores and transferability of those scores. And then lastly, the pharmacogenomics angle, we really want to get a better understanding of variants that are affecting drug response across these populations. Kind of a lot, tying this into the kind of the culture of the country and the healthcare ecosystem, of the company and the healthcare ecosystem that we're trying to build. We want to be ethically responsible, and we want to create jobs, transfer knowledge, be environmentally aware, but also build, prevent brain drains and things like that, and build kind of a within continent empowerment of science. And we want to continue to invest in research infrastructure and just expand, even beyond genomics into just kind of transforming healthcare on the continent and sharing the benefits with the local communities. So, thanks, and there are many people involved here, specifically in my team who carried out, some of the kind of the PopGen work that I presented, and then the 54 gene leadership and really study participants, collaborators, the entire 54 gene team. And please reach out if any questions. And here's a comment from Abasi in Nature Biotech yesterday, which is quite timely. And I'll leave it up there for folks to read. But yeah, thank you very much for listening, and I welcome any questions.
Thank you so much, Colm, for such a brilliant insight into the activities that 54 Gene is doing and into your own work. And I open the floor to questions and comments. I see a first comment here from Driti. Perhaps it's just a point of order. Driti says, great talk. And she says, that referring to all ethno-linguistic groups as tribes is not common in African population genetics and probably should be done with caution. I think it's just a point of order there.
Oh yeah, good to know. Yeah. So the preferred nomenclature is ethno-linguistic groups.
Yeah, and then we have a question from Precious. She said, Precious has a couple of questions. So Precious, would you like to unmute and ask your questions?
Right, thank you. Thank you very much, David. And thanks a lot for the content. It was very interesting getting to know more about 54 Gene and all about genomics in Africa. The first question I wanted to ask was on 54 Gene. I think someone answered that in the comment, Delali, as regards, because I saw a slide that said that the organization works across seven states, and I was curious because outside Lagos, I wasn't aware of other states where the organization worked in, and I was curious about those states.
Yeah, so I think Delali has answered in the thread, right? It's 6 different regions. Delali, yeah, I mean, I think we're in most states at this point. I can't really speak to the specific regions, but I think if you kind of reached out to us, we could kind of give you a better breakdown on that. I don't know off the top of my head.
Oh, okay, no, not a problem. And the second question, there was this slide that made reference to the community-based, sorry, the portfolio of hospital-based studies and spoke about, working with a lot of tertiary hospitals and fewer primary healthcare facilities. I mean, I understand that the diagnostic capability that is resident with 54 gene would be utilized mostly by tertiary facilities. But I was wondering, given that PHCs are closer to rural populations, how do you try to inculcate these rural populations, with these PHCs in your studies?
If I got that, how do you try to, you faded out, how do you try to engage with the rural communities, or what was that again?
Can you hear me better now?
Yeah, I can hear you better now. Yeah, go ahead. Sorry.
Yeah, so I was asking, given that primary healthcare facilities are closer to rural populations, how do you try to engage with rural populations for like hospital-based studies, since there's more, it seems like there's more engagement with tertiary institutions?
Yeah, I mean, I think that's kind of a question for the outreach strategy and education and engagement, community engagement. I mean, it's a very open-ended question.
Perhaps I can try and,
yes,
can you guys hear me?
Yeah, hi Delali. Yeah.
Hi everyone. Hope everyone's doing well. So there are really two types of ways we engage as far as recruitment is concerned. So one, to your point earlier on, Precious, in terms of your question, is around being able to do our hospital-based recruitment, where we do work with quite a few tertiary institutions across Nigeria. But we also do community-based recruitment as well too. And so working in certain catchment areas that allow us to be able to get into rural populations and work with those community leaders, we do have ways for which we engage from a community recruitment perspective and working within certain catchment areas to make sure we have representation from the rural side as well. Hopefully that answers your question. Are you still there?
Yes, very much.
Yes, thanks a lot, Delali, and thanks, Colm, as well. I don't know, Colm, whether you wanted to add anything to.
No, Delali got it. I mean, Judith had a question about the relationship between Regeneron and 54 gene. Yeah, no, there is none. I just worked in Regeneron in the past.
Yeah, there's no relationship there. I think.
Great. Then we have a question from George. George.
Hi there.
Hi, George.
How's the sound?
Oh, hi, George.
Hi there.
Hi,
hi, Colm. Thank you for an excellent talk. It's really exciting to see the work. I have two questions. One, I saw that there was not much done in the African South. Are you planning to do more work in Southern African populations?
Yeah, yeah, we definitely would love that. And I think it's just a matter of building that research network. And I think was it you who presented, or I saw a presentation on PopGen in South Africa on this webinar series,
Yeah.
and it looks like there's really kind of fascinating structure there too. So I think hitting South Africa would be incredibly valuable and important. Yeah.
Yeah, excellent. And the second question is a follow-up from our work that we did before, is that as you sequence more African populations, you'll find a lot more, not just common variants, but rare variants, which may have really strong effects. I saw the plan to use a chip to try and get more data in, but those rare variants might still elude you. Do you have any ways to get around that problem?
Yeah, I mean, yeah, sequencing really, right? But the chip, so, we haven't really run those experiments yet where we try to build as good a reference, like reference, imputation reference as possible. I know you can get pretty rare with a big reference panel. So, in theory, if we had 25 to 50,000 whole genomes, I mean, I think we would get pretty rare samples. But no, you're right. I mean, I think locus discovery, if we found a locus with the chip and then did a kind of a fine mapping, targeted sequencing approach, we might get the rare variants that way. It's a bit more cumbersome. It's nicer to just blanket the entire genome. But there are some of the strategies, but yes, welcome any other ideas you might have.
Yeah, well, I think we'll have to keep talking in the future. It's really exciting, and I think a lot more sequencing and work to make it happen.
It's great. Thank you, George.
Yeah, cheers.
Thank you, George. Hussam?
Thank you, David. Thank you, Colm. That was really a very interesting talk. So I have a really maybe broad question here. So most of the African countries, they're struggling now to implement precision medicine healthcare system. And I imagine also that's not the problem only of Africa. All over the world, I think even some European countries, they have the same problem. So my question is, how do you keep your activity awake in this kind of environment? Is it something relative to high demand on precision medicine, or is it a particularity in your business model? So I'm really curious to know more about that.
Yeah, no, I think it's just a desire to gain a better understanding of the negative effects of, of naturally occurring variation on outcomes in hospital systems, and I think that will benefit the, the systems and the communities we interact with. It's not a kind of an explicit aim, but I think it's a desire to globally really to build a better understanding of these things, and even in clinical trials, sampling more diverse populations to kind of better characterize the kind of potential adverse outcomes. It's not enough anymore to just run a clinical trial in a homogeneous genetic group and to assume that approval would expand globally. I don't know, does that answer, does that help? Or do you want more?
Yeah, a little bit, yeah. Yeah.
Thanks a lot, Hussam. Sami?
Hi, Colm. It's a great talk. It's a pleasure to be here. I'm here on behalf of Kaizen, and it's really a pleasure to be a part of this talk. My question to you is that, the bioinformatics part of such a massive project like this is very challenging, and it's a bottleneck to many projects that I work with in the region. What would you say the main lesson that you took away from managing such large amount of data, something that you learned working as part of 54Gene in terms of managing the massive amount of data and the infrastructure involved in 54Gene? Just curious if you've learned sort of a theme or a takeaway message in this particular project.
Yeah, I think it's still early days, and I suppose we're still at a reasonably small scale. It could really, that scale could get much bigger. Some of the things we've noticed is, internet connectivity can sometimes be a bit dodgy, and uploading data to the cloud. Once you get the data in the cloud, we can do a lot, and we can spin up more nodes for compute, and we can really analyze big data sets in a very short period of time. But getting the data to the cloud is a potential bottleneck. So anything that could increase stability and speed of uploads is really important. I remember, I think back in Regeneron we had to build specific new cables for higher data transfer rates to get things into the cloud. So I think this is a common problem with big data.
And just maybe another follow-up question on one of the points that were raised by one of the attendants is, for low variant allele frequency, have you considered digital PCR because of the lower limit of detection can get to about 0.001%? Would that be of interest?
Yeah, definitely of interest. We haven't kind of expanded into all those new technologies just yet, but I think that's a fantastic technology, and we'd certainly be happy to explore it. Yeah.
Right, because some the targeted DNA panels lower limit of detection is around 0.5%.
Yeah,
but it's fascinating with the digital PCR technology. They can get up to 0.001%, which is on the single molecule level, which is quite fascinating.
That is fascinating. I mean, I think some of the low-hanging fruit would be like variants that are ultra rare in European populations, but actually higher frequency. And so I think we could have a lot of luck in detecting, globally rare variants that are actually locally not so rare. And I think once that low-hanging fruit has been explored, and you're getting globally rare, so rare in like European and rare in African population variants of interest, that technologies like that would be critical. Yeah.
I have a question.
Thank you.
Yeah. I have a question from Olaitan.
All right, thanks. Let me just be very fast. So my first question is if 54 Genes has immediate plans for scale up to other African populations that are currently underrepresented in your work, and then when you collect these samples, what specific samples do you collect? So I'm just curious what you're collecting and what kind of metadata do you collect at the time of getting the samples?
Yeah, I mean, I think that kind of varies by country, and ethics approval, and what the study design is and approval for that study is. But it could be FFP and blood and saliva and kind of various tissue types depending on the nature of the study. But it really depends on how what the country is and what the guidelines and ethics.
You just extract DNA from the samples, and that's all.
Yeah, I mean, yes, that's one of certainly the primary focus right now. Yeah.
Okay, and then the metadata you get with it.
Well, the metadata would be really depending on how the study is calibrated. And so, if we're running a study where we're collecting samples and phenotypes, then the metadata would be phenotypes and would be kind of a lot of different phenotypes or a specific targeted phenotype. But it really depends on the nature of the study. But you really would want some metadata that would allow you to do a reasonable association scan.
Correct, because I'm curious, I mean, when you get these variations, you want to know the impact, right? And if you don't have enough metadata information that would help you characterize what the variations mean, it might be hard to tell.
Oh yeah, well, you can annotate the variants that once you've kind of sequenced, and you can detect, the functional classes of the variants. But I mean, you do need things like age and sex and things that are kind of confounders to be able to run a kind of controlled association scan.
Okay.
Yeah.
So your immediate plans for scale up to other African populations, because you're just in Nigeria, right?
Yeah, Nigeria, and as I mentioned, I think three other countries. I mean, there are always ongoing efforts to continue to expand and to work with communities and governments to continue to expand sequencing on the continent. Yeah.
Thank you.
Sure.
I see Delali, you want to clarify on that?
Yes, no, I was just going to add to what Colm just said, but it's correct. So we are, so I did see a question in the chat as well too. So we are expanding our efforts. And I think Colm did share a side to that effect. Primarily, we've had a huge operational base in Nigeria, but we are working with other academicians and other partners as we look at other areas to expand across Africa. So that's ongoing at the moment. I also did just want to quickly address, I believe there was a question from Pharaoh. Hopefully I've pronounced your name correctly. Apologize if I didn't. But yes, we do have internship programs that we do run. I don't believe we have any active at the moment, but later on this fall, we will be opening up some additional internship opportunities. So you can probably keep posted to our page on that. And then I believe there was a question around ethics or ethical consent, which, yeah, we do get broad consent. We also get ethics when we're working with institutions, we get it at both the institutional level as well as the national level. And that's one of the key parameters for us being able to do anything related to our studies.